Thank you for giving me the opportunity to present my work at this forum. So yeah, this is Haseeb Manzoor. I'm a PhD scholar at the Segata Lab, University of Taranto, Italy. And today the topic of my research is metagenomic analysis reveal E. coli-derived siderophores as potential signatures for breast cancer. So this is the work that I conducted at National University of Sciences and Technology, Pakistan. Moving forward with the introduction. So, this is a healthy gut, which is influenced by a healthy lifestyle. And once we adopt a healthy lifestyle, this improves our overall health. We see that there are numerous ways that this gut actually communicates with human body, and one of them is with the production of compounds known as the metabolites. These metabolites, they do a very important job in the human body as they can regulate energy metabolism. They are involved in immune system modulation, nervous system regulation, and are essential for vitamins and compounds production. But what happens if this healthy gut get dysbiotic due to factors like genetic factors or poor lifestyle? This has severe consequences of a human body as it can result in several diseases like multiple sclerosis, diabetes, or cancer. So over the past few years, there is a lot of buzz in the scientific community to manipulate the gut microbiome to prevent diseases. And in our case, what we were doing, we are exploring that in terms of breast cancer. So our question was that could the manipulation of gut microbiome would have any therapeutic potential for breast cancer? And for that, we referred to the literature review. And after doing an extensive literature, we have come up with the problem statement that the research gap in the study of gut microbiome metabolite association in breast cancer patients require further exploration. And our proposed solution was that the shotgun metagenome analysis could be used to identify specific metabolites produced by the gut microbiome that are associated with breast cancer risk and potential therapeutic implications. So, moving forward with the objectives, we had to analyze gut microbiome composition in breast cancer patients using the shotgun metagenome sequencing data, to identify secondary metabolites with potential anti-cancer activities in the gut microbiome of breast cancer patient, and lastly, to establish correlations between specific microbial taxa, metabolites, and breast cancer using the machine learning models. Moving forward with the data, we had a secondary data which we got from sequence read archive. We had 131 metagenomic samples that were distributed among premenopausal cases and controls, and postmenopausal cases and controls. Moving forward with the methodology, we got the raw metagenomic reads from the SRA database, as I discussed earlier. Further, we performed the quality control via FastQC, FastP, and BBDuct, and out of that, we got high quality metagenomic reads. Now, these metagenomic reads, we got further, we performed the metagenomic assembly via MegaHit, and out of that, we got taxa profiling of context via M4R2, and metabolites identification was done via anti-SMASH. Further, the abundance estimations of the taxa as well as the metabolites were performed via cover M tool. Now, the abundance matrices of taxa as well as the metabolites were further supplied along with the metadata about what kind of phenotype was there. It was further supplied to three further analysis: the correlation analysis via Pearson correlation, association analysis via Mazarin 2, and machine learning. In the machine learning format, what we did, we basically used initially six machine learning classifiers like random forest, logistic regression, support vector machine, extreme gradient boosting, k-nearest neighbor, and gradient boosting. And out of six models, we took the top three best performing models and we classified our samples based on the microbial and metabolic features. And through that, we captured some highly accurate metagenomic features that were associated with breast cancer. So out of these analysis, we got the breast cancer associated taxa as well as the breast cancer associated metabolite. Then we perform iron metabolism analysis. Now, why we perform iron metabolism analysis, I'll talk in the later slides. So moving forward with the results, we perform the taxa association analysis. The premenopausal cases versus control. In premenopausal cases versus control, what we saw that E. coli, it showed significant positive association with the premenopausal cases. While on the other hand, in postmenopausal cases versus control, we saw that E. coli, it again showed significant positive association with the postmenopausal cases. Now, if you see that regardless of the menopausal status, E. coli showed significant positive association with the breast cancer cases, which led us to deduce that E. coli might be having some underlying mechanism that is basically causing the breast cancer to progress. So that needed to be further elucidated. Or on the other hand, our ensemble classifier, which captured the microbial signatures and classified based on that the premenopausal cases and controls as well as the postmenopausal cases and controls with an accuracy of more than 90% and AUC of more than 95%. And once getting the what are the high ranked taxa out of these ensemble model, we saw that E. coli along with Klebsiella pneumoniae and Bacteroides fragilis, it again topped the chart. So it basically validated or strengthened our previous results. Moving forward with the metabolites association analysis, we saw that siderophore and thiopeptide, these were the two metabolites in premenopausal cases versus control. They showed significant positive association with the premenopausal cases. On the other hand, these siderophore again they showed significant association along with thiopeptide in postmenopausal cases, which led us again to interrogate that what would be the underlying mechanism of these siderophores or thiopeptides that could be having a role in the breast cancer progression, because regardless of the menopausal status, they were highly associated with the breast cancer cases. So again, strengthening the previous result, we saw that ensemble classifier, which was using the metabolic signatures, it classified the premenopausal cases and controls as well as the postmenopausal cases and controls with an accuracy of 90% and AUC of more than 95%. And we again saw that siderophore and thiopeptide they topped the metabolite ranking chart. So which also strengthened over previous result. Moving forward, just to give you a flashback that out of breast cancer associated taxa, we got E. coli, and out of breast cancer associated metabolite, we got siderophore. Now, what could be the mechanism between these two? To identify that, we referred back to the literature review. And through literature, we got some striking figures that up to 40% of the breast cancer patients are anemic. And once the patient is already facing the iron limiting condition, another research reported that it increases the angiogenic potential of the breast cancer patient, which increases the tumor size. Now, if we see that on the other hand, the dysbiotic gut has also been producing a bacteria which is known as E. coli. Now, E. coli, it also needs iron for its survival. Now, what happens, it creates a competitive environment between the host as well as the bacterium to get the iron. Now, at that critical point, what this bacterium does, it releases compounds known as siderophore. Now, these siderophore, they are very crucial at that point. What they do, they're basically the iron chelating agent. They go to the host, takes the iron, and comes back to the bacterium, ultimately leaving the host into more iron scarce conditions, and it increases the angiogenic potential, facilitating and further increase in tumor size, and hence there is tumor progression. So, out of that, our hypothesis was that the iron acquisition in breast cancer by E. coli is driven by siderophores that are as part of the iron metabolism pathway. And our extended hypothesis was that the genes responsible for iron metabolism in E. coli will likely show a positive association with the breast cancer cases. And once carrying out our analysis, we saw that 25 iron metabolism genes were identified, which has role in several processes like heme biosynthesis, siderophore mediated iron acquisition, iron delivery, intracellular transport, iron homeostasis or metabolism, and iron storage. And once we performed the association analysis, we saw that all genes except heme they showed significant positive association with the breast cancer cases, which validated our hypothesis. Moving forward, this study concludes as it provides a connection between the gut bacteria and metabolite, which was once a gap in terms of E. coli and siderophore via the iron metabolism pathway. This could be a new detection method for breast cancer, and it provides a new avenue to treat the breast cancer. So yeah, thank you so much. That was all from my side.